Okay, so this will be the last lecture on exam 1, right? Is plasma membrane and membrane potential. The last bit of information that we're going to cover for exam 1. So what we're going to do is we're going to talk about the structure of the plasma membrane. We talked about very briefly when we talked about cells. We're going to talk about it in way more detail today. Then we're going to set up a membrane potential. And this is really key. You need to understand membrane potentials for exam 1. You need to understand membrane potentials for exam 2. You need to understand membrane potentials for exam 3. Get a little break on exam 4, and then you'll need to understand them for the final as well. Okay, so if you don't get membrane potentials, you are going to miss loads of quiz and exam questions on multiple quizzes and exams. This is a really key point. So ask questions when we cover it and make sure you understand it. Okay, because it's super important. If you don't understand it, you're going to miss lots of points. Okay, and then we'll talk about transport across the plasma membrane. Unassisted versus assisted. Okay, so assisted just means transporters involved. Right, so our plasma membrane, which is also known as the cellular membrane, is a really thin layer of lipids studded with proteins. Okay, and it surrounds every cell in your body. Okay, every single cell in your body has a plasma membrane or a cellular membrane. Okay, and the plasma membrane, because it's made up of lipids, is semi-permeable, meaning it's not freely permeable to everything. So because it's made up of lipids, do you think it's permeable to hydrophilic or hydrophobic things? Hydrophobic. Okay, so only hydrophobic molecules can freely cross. Anything that's hydrophilic has to be transported. Because it's transported, that means it can be regulated. So this means cells, the intracellular fluid, can have a completely different makeup than extracellular fluid. Okay, because of the semi-permeable nature of the plasma membrane, your cells can control what's inside versus outside, if we're talking about hydrophilic substances. Okay, the plasma membrane also participates in joining cells, okay, to form tissues and organs. And then it also plays an important role in allowing the cell to respond to changes in the cell's environment. Okay, most hormones are hydrophilic. Okay, so the steroid hormones are hydrophobic and they cross the plasma membrane. And then thyroid hormone is also hydrophobic, so it can cross the plasma membrane. But most other hormones are hydrophilic, so that means they never enter the cell. They just bind to receptors on the extracellular side. For example, insulin. Right? When does your pancreas release insulin? Due to sugar. Due to an increase in sugar or a decrease in sugar? Increase, okay? So blood glucose levels start to rise, pancreas releases insulin. Insulin binds to receptors on insulin-sensitive cells. Insulin-sensitive cells are just cells that have receptors for insulin. Okay, so that would be skeletal muscle and hepatocytes and adipocytes that store fat, hepatocytes in the liver. Okay, when they bind to insulin, then they start to absorb the glucose. Okay, so those cells are responding to their environment, i.e. an increase in blood glucose, via their plasma membrane, i.e. a receptor that is inside the plasma membrane. Okay. So plasma membrane is what's called the fluid mosaic molecule. It's a phospholipid bilayer. Okay, so those phospholipids, and when we look at it, the proteins inside the plasma membrane are not rigid. Okay, so that's what that fluid mosaic model is. Okay, there's fluidity. Things are always moving around. Okay, but because of phospholipid structure, and the fact that there's a polar head region and a non-polar tail region, right? These phospholipids spontaneously form a bilayer or a double layer, okay, where the polar head regions interact either with intracellular or extracellular fluid, and the non-polar tail regions interact with each other. Okay, so the interior of the plasma membrane is non-polar, and then you've got the polar head regions that are forming the boundary with intracellular fluid and extracellular fluid. Okay, phospholipids are amphipathic, polar and non-polar. Right, so here we've got phospholipids. So we've got the fatty acid tails interacting with each other, right, via hydrophobic interactions. And then we have the polar head regions interacting with water in the extracellular fluid, polar head regions interacting with water in the intracellular fluid. Okay, and then that interior portion of the plasma membrane is hydrophobic. So it serves as a barrier. Only hydrophobic substances can freely cross. So what's an example of a hydrophobic substance? Mentioned one class already. Fat or steroids. Okay, steroid hormones can freely cross the plasma membrane. Okay? Oxygen and carbon dioxide are also hydrophobic. So they're never transported. They're just going to move down their gradients. So oxygen's going to move through the plasma membrane into the cell, and then through both membranes surrounding the mitochondria into the matrix. And what happens to it in the matrix? What happens to oxygen?
Electron transport chain receptor.
Not receptor, what is it?
It's the terminal receptor.
Terminal, not receptor, acceptor. Acceptor, okay? So it's the terminal electron acceptor in the electron transport chain. Okay, but it can freely cross all lipid bilayers, including the cell membrane as well as the mitochondrial membranes. Yeah, but did you have a question?
So you said oxygen. What was the other example?
Carbon dioxide. Oxygen and carbon dioxide are non-polar molecules that freely cross plasma membranes. Okay, they are not particularly water soluble. That's why we have a carrier protein in our blood for oxygen, right? Because if we were just depending on dissolved oxygen in our bloodstream, we wouldn't have enough oxygen to give to our cells. Okay, so we have hemoglobin inside our red blood cells to carry the oxygen, which is nonpolar. Okay, this lipid bilayer, those lipids are also really fluid. When we talked about lipids, what kind are you supposed to eat? Saturated or unsaturated? Unsaturated. And why are you supposed to eat more unsaturated? Because you can true, because you can't unsaturate fat, but you can saturate it. But which one's more fluid? Unsaturated, right? Okay, so our plasma membranes are selectively permeable. Non-polar substances freely cross oxygen and carbon dioxide. You could add steroid hormones to this as well. Polar substances cannot freely cross. And that means they can be regulated. And that means you can have different concentrations of polar substances in intracellular fluid versus extracellular fluid. You need to memorize as soon as possible, okay, where potassium, sodium, calcium, and chloride are in higher concentration, intracellular fluid or extracellular fluid. You don't have to memorize the exact numbers, right? The exact millimoles in intracellular versus extracellular. Okay, so ICF is intracellular fluid, fluid inside your cells. ECF is extracellular fluid, fluid outside of your cells. So we'll just take this opportunity right now to draw a cell and put our key ions. Okay, so I want you to memorize where sodium, potassium, chloride, and calcium are in higher versus lower concentration. Okay, so where is sodium in higher concentration? Extracellular or intracellular? Extra. Okay, so I'm going to put a big Na positive in outside of my cell and a little Na positive inside of my cell. Okay, where's potassium in higher concentration? Inside. And why is potassium in higher concentration inside and sodium in higher concentration outside? Anybody know? We haven't talked about it yet, but if you know, that's awesome.
I believe, isn't that how glucose enters the cell?
That is true. Glucose enters the cell using the sodium gradient. Yeah.
And isn't it something like, as part of that, potassium gets concentrated? I don't know the full.
So there is a transporter, an active transporter called the sodium potassium pump on all of our cells that uses ATP to pump 3 sodium out, 2 potassium in. Okay? So our cells expend energy to kick sodium out and bring potassium in. Right? Our cells are regulating the amount of sodium and potassium by using ATP. Okay? So our mitochondria are churning through all that glucose and all those fatty acids and carbon skeletons of amino acids in order to make ATP to do membrane transport. Okay? And we'll talk about that sodium potassium pump at the end of this lecture. Okay, but your cells are constantly kicking sodium out and potassium in order to maintain differences in ion concentrations. Okay, chloride. Chloride's an easy one because it's sodium's best friend. So if you memorize where sodium's in higher concentration, then just put chloride in that same place. Okay, so just remember sodium and chloride are best friends. So where's chloride in higher concentration? Outside the cell. Right, that makes it in lower concentration inside the cell. Okay, now calcium. Where's calcium in higher concentration? Outside the cell. Okay, and the reason calcium gets 2 pluses is because it's a divalent ion. It's given off 2 electrons. Okay, so that's why it gets 2 pluses. And then less calcium in intracellular fluid. Okay, what I want you to notice is look at extracellular fluid concentration of calcium versus sodium. Which one is in way higher concentration in extracellular fluid, sodium or calcium? Sodium, okay? Sodium is the major extracellular cation. Okay, an ion is anything that's charged. So anything that's taken up or given off an electron. A cation is positively charged, so it's given off an electron. So that's sodium, potassium, and calcium. And then an anion has taken up an extra electron, that's chloride. Okay, what's the major intracellular cation? Potassium, right? It's the major intracellular cation. All right, what I want you to also notice, if we look down here, amino acid concentrations and protein concentrations, much higher in intracellular fluid than in extracellular fluid, which should make sense. Okay, your cells are filled with proteins because proteins are doing work. Building blocks of those proteins are amino acids. Okay, but those proteins and those amino acids are negatively charged. So just remember that. Right, when we talk about membrane potentials, that will come into play. All right. So, don't have to memorize the exact numbers. You do have to memorize where they are in higher concentration versus lower concentration. So for sodium, potassium, chloride, and calcium. Okay, and just memorize it. I promise it will pay off. Okay. Right, so here we have the plasma membrane being shown with proteins for the first time. So each one of these gray circles and the 2 squiggly lines is a phospholipid. And then all these purple blobs are proteins. Okay, so you remember the plasma membrane is a fluid mosaic molecule where we have a phospholipid bilayer studded with proteins. Okay, and some of those proteins go all the way through the plasma membrane. Some of them are only found on the intracellular side. Some of them have carbohydrates attached to them and would be a glycoprotein. Some of the lipids have carbohydrates attached to them and would be a glycolipid. And then notice here, we've got cholesterol that's inserted itself in between those fatty acid tails. Because is cholesterol hydrophilic or hydrophobic? It's hydrophobic. It's a type of fat. right? So it's associating with those fatty acid tails. And cholesterol adds stability to the plasma membrane, right? And it can also enhance fluidity to a degree. But you tend to find it where you see lots of proteins, okay? Because the proteins, you want the membrane to be a little more stable there. Okay? Some of these proteins might be receptors, like an insulin receptor would be a protein. They might be a transporter for ions, for glucose, etc. They might attach to the cytoskeleton to help give structure, right? Those proteins play a lot of roles. Okay, so we've got cell surface proteins. Those are just found on one side of the cell, and then we have transmembrane proteins that are inserted within the plasma membrane. Okay, cholesterol gets tucked between those little yellow molecules on the previous slide, and it contributes to fluidity by getting in between those fatty acid tails. It also stabilizes the plasma membrane, mainly for those proteins, to help keep those proteins in place. Okay, and then a little bit of carbohydrate as well. But carbohydrates on the outer surface only. Okay, so carbohydrates that are associated with the plasma membrane are on the outer surface only. And they're marking the cell as a particular type of cell, so their self-identity. So that early on in development, when cells are trying to figure out where to go to form tissues and organs, etc., these self-identity molecules are really important. Right? The Rh factor on your red blood cells determines if you have positive or negative blood, is also a carbohydrate. Okay, so let's talk specifically about those proteins. We've got these integral membrane proteins that actually get embedded. If they face the intracellular side, they're often doing some sort of chemical reaction in the cytosol, right? And G proteins are examples of these. We haven't talked about G proteins yet, okay? But if you've taken cell bio, you know all about G proteins, okay? So G proteins will start an enzymatic reaction inside of the cell. Okay, extracellular facing, so these are just showing on the extracellular side. They're going to catalyze the reactions in extracellular fluid or act as receptors. Okay, so receptors for peptide hormones are extracellular facing. So they're going to have what's called a ligand binding site to bind to that particular compound. Anything that binds to anything that binds to a receptor can be called a ligand. Okay, and then transmembrane proteins go all the way through, and they can serve as channels for ions or water, okay, or they might serve as carrier molecules. And then we get some of these peripheral membrane proteins that are really loosely bound, and they usually are attaching the cytoskeleton to the plasma membrane. Right? So you're going to just find them on that intracellular side. Okay, any questions about the structure? So you need to understand how phospholipids are amphipathic and form that lipid bilayer, and then that there's other things in there besides phospholipids, things like proteins and cholesterol and a little bit of carbohydrates, but only on the extracellular side. Okay. Are you guys ready to set up a membrane potential? Again, super important point of the class. Okay, so if you don't get it, you need to make sure you're understanding this for exam 1, 2, and 3. Yeah?
I just have a quick question on peripheral membrane proteins.
Yeah.
What's their function?
So the peripheral membrane proteins, they're mainly there to attach the cytoskeleton to the plasma membrane.
Oh.
Yeah. Okay. Any other questions before we go on? Okay. Okay. So, in order to understand membrane potentials, we need to understand that ions move down both concentration and electrical gradients. Okay, so ions move down both concentration and electrical gradients. Uncharged molecules only move down their chemical or concentration gradient. Okay, ions, because they are charged, are going to move from where they're in high concentration to low concentration. But they also want to move to the opposite charge. Okay, so positively charged ions, i.e. cations, want to move towards anions. Anions want to move towards cations. And this is why sodium and chloride are best friends. Sodium's the cation, chloride's the anion. Okay, but sodium wants to move towards any negative charge. Chloride wants to move towards any positive charge. Okay, so we've got chemical gradients to think about and electrical gradients to think about now. Okay, our cells are charged. Our cells have an unequal charge separation across the membrane, right, which is called membrane potential. And the reason why it's called membrane potential is because it's a source of potential energy. Okay, our cells can use the membrane potential to do work. One of those things might be transporting glucose. Okay. Our neurons use the membrane potential to send signals. Right? So the membrane potential is present in all our cells. So the unequal charge is that extracellular fluid has a net positive charge and intracellular fluid has a net negative charge. Okay? And what do you think is giving the net positive charge to extracellular fluid? What's the major extracellular cation? Sodium. Okay? The net negative charge in our intracellular fluid are these intracellular anions, which I denoted as a capital A with a negative symbol. Those are the proteins inside our cells. Remember, we have way more proteins intracellularly than extracellularly. They're trapped. Okay. Right. Chemicals that are uncharged will move just down their concentration gradient. Okay, so here we have a chemical that's in higher abundance in intracellular fluid versus extracellular fluid. So we could think of this orange circle as carbon dioxide. Okay, so carbon dioxide is in higher abundance inside our cells than outside of our cells. So carbon dioxide is going to move down its chemical gradient to the extracellular fluid. In panel B, we can think of the orange circles as oxygen. Oxygen's in higher concentration in extracellular fluid than intracellular fluid. So it's just going to move into our cells down its chemical gradient. Okay, both oxygen and CO2 are not charged, so they just move down their chemical gradients. Okay, charged molecules also move down electrical gradients. Okay, so our cells have an unequal distribution of anions and cations across the plasma membrane because charged molecules can't freely cross the plasma membrane. Okay, so the cell can regulate where they are. Okay, membrane potential is denoted as a capital V with a subscript M. Okay, so that's V subscript M is membrane potential. All of our cells, if they are living, have a charge associated with them. Right? They all have a membrane potential. And they can use that membrane potential to do work. I already defined all of this, but let's do it again. An ion is anything charged. So if it's given up an electron, it's a cation. And if it's taken an extra electron, it's an anion. Okay, so here we have the plasma membrane, and we have intracellular fluid versus extracellular fluid. There is a net negative charge in intracellular fluid. That doesn't mean there aren't any cations. And in fact, what is a cation that's in higher abundance in intracellular fluid than extracellular? What's the major intracellular cation? Potassium. Okay, so you can think of all these little purple circles as potassium. There's just more anions than cations. Okay, so the net charge is negative. Extracellular fluid does have anions. What's the major extracellular anion? Chloride, right? But it just has way more cations than anions, so it gets a net positive charge. Okay, so we have a separation of charge across the plasma membrane. Okay, so our ions care about that charge. Okay, positively charged cations want to move towards the anions inside the cell, and negatively charged anions want to move towards the cations in extracellular fluid. Okay, so anything that's a cation wants to get in, anything that's an anion wants to get out. Okay, so let's now think about both electrical and chemical gradients for our key ions. Okay, so we have our cell. We've got net negative charge inside, net positive outside. So we've got the charge separation. And then we've got high sodium outside, high chloride outside, high calcium outside, and then high potassium inside. These anions that I put in here are trapped. They can't move. So we don't care about their gradients. They're not going anywhere. Okay, but let's think about sodium. We're going to use solid lines for a chemical gradient, and then dashed lines for an electrical. Okay, so sodium, chemically, which way does sodium want to move? With words, into the cell or out of the cell? Into the cell. Okay, so chemically sodium wants to move into the cell. So I draw my solid arrow pointing towards the interior of the cell. How about electrically, which way does sodium want to move? Into the cell. Okay, so I draw my dotted line into the cell. Okay, so this means sodium's electrochemical gradient, so that's the sum of both the electrical and chemical gradient, is into the cell. So sodium really, really, really wants in. And the fact that sodium really, really, really wants in, the cell can use to pull something else up its concentration gradient. Okay, and we'll talk about that when we talk about active transport. Okay, how about potassium? Chemically, which way does potassium want to go? Into the cell or out of the cell? Out. Okay, so my solid arrow is pointing out. How about electrically, which way does potassium want to go? Into the cell or out of the cell? Into. Okay, so this means potassium has opposing electrochemical gradients. Chemical always trumps electrical. Okay, so that means if given the chance, potassium will leave the cell. It won't rush out though, because the electrical gradient is holding it in. But it will slowly leak out of the cell, because the chemical gradient is stronger than the electrical gradient. Okay, let's do calcium. Which way does calcium want to move chemically? Into the cell. Which way does calcium want to move electrically? Into the cell. Okay, how about chloride? Chemically, which way does chloride want to move? Into the cell. And electrically, which way does it want to move? Out of the cell, because it's an anion. Okay, and just like potassium, the chemical gradient is stronger than the electrical. So if given the chance, it will enter the cell. Okay, you guys understand how to set up chemical and electrical gradients? So this is why I said memorize where the key ions are as soon as possible, because once you memorize where they are in higher abundance, you can set up their chemical and electrical gradients, which I recommend doing on your piece of scratch paper you get to have for the exam, right? You can make your cell, set up the chemical electrical gradients before you start answering questions about it. Scratch paper, not a cheat sheet, okay? So here we have a figure showing what we just did. Okay, so the electrical driving force for potassium is into the cell. Chemical is out of the cell. And the thickness of these arrows denotes how strong those forces are. So the yellow arrow is fatter than the blue arrow. That means the chemical gradient is greater. than the electrical one. So the net electrochemical driving force, shown in the green arrow, is to leave the cell. Now we have some numbers underneath the cell. What's VM again? Membrane potential. Okay, so this is the first time we're seeing membrane potential of most of our cells at rest is minus 70 millivolts. Okay, membrane potential is always the charge inside the cell. Okay, so inside of our cells, the charge is minus 70 millivolts. So we've got net negative charge inside our cell. If you have a membrane potential of 0, that means there's no charge distribution. That's a dead cell. Okay. This other number is the equilibrium potential of potassium. Okay, so E is for equilibrium, the K is for potassium. What this equilibrium potential means is that if a cell becomes freely permeable to potassium, this is what the membrane potential would be, minus 94 millivolts. Okay, the membrane potential would drop if our cell becomes 100% permeable to potassium. Right? And the reason why membrane potential would drop is because cations are leaving the cell. Right? Because the electrical chemical gradient is to leave the cell for potassium. Yeah?
Do we need to know the equilibrium potential for potassium chloride?
You want to know that the membrane potential for our cells is minus 70 millivolts, and that the equilibrium potential for potassium is lower than that. Mm-hmm. Yep, but you definitely want to know membrane potential is minus 70 millivolts, and equilibrium potential for potassium is lower than that. Yes?
Minus 70 is when the cell is at rest.
When the cell is at rest. Excellent, yes. Minus 70 millivolts is for cells at rest. Next week, we're going to talk about neurons, which are excitable cells. Right? And they're sometimes not at rest, and their membrane potential differs. Okay. If you really understand membrane potentials well and how cells set them up, the reason why I'm making such a big deal about this, it makes action potentials so easy. Okay? You'll actually understand them rather than just memorize facts about them. And you guys all want to understand everything, not just memorize it, right? Okay, so membrane potential is a difference in charge across the plasma membrane, and that negative charge is due to those fixed anions, okay, which are proteins. Other ions are diffusible and therefore can cross the plasma membrane because there's channels for them. One of those diffusible ions is potassium. Okay, potassium can cross the plasma membrane, right? So it's going to slowly leak out down its electrochemical gradient. Sodium is not diffusible. We don't have sodium channels that are open in cells at rest. Okay, and in fact, we have a sodium-potassium pump that's constantly pumping sodium out. So last thing we're going to do with our cell up here is we're going to put an X through calcium because it's not diffusible. So everything I put an X through means it's not diffusible. Okay, so calcium's not diffusible. Potassium is, so I'm not going to put an X. Okay, sodium is not diffusible, so I put an X. And then chloride is not diffusible, so I'm going to put an X there as well. Okay, so at rest, potassium is the ion that can slowly diffuse out of the cell down its electrochemical gradient. Okay, so all of our cells have a resting membrane potential, right? And most cells, it sits at minus 70 millivolts because of those non-diffusible anions, i.e. proteins that are stuck inside of our cells. What we're going to talk about next week is that nerves and muscle cells are excitable, and they can change their membrane potential when excited. Okay? We're going to watch a video as the last thing about how cells set up resting membrane potentials. And it uses neurons as the example, but all of your cells do the same process for setting up a resting membrane potential.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Right, so we're plasma membrane. today, right? We're going to separate it into two types of transport: unassisted and assisted transport. Right, so we talked about the plasma membrane on Monday, and that it's a phospholipid bilayer that's studded with proteins. And that phospholipid bilayer makes the plasma membrane selectively permeable. Right? What can freely cross that lipid bilayer? What can dissolve in the fatty acid tails? Hydrophobic substances. Right? So substances have to be hydrophobic in order to freely cross the plasma membrane without any sort of transporter. Okay? Lots of things can cross the plasma membrane, they just involve transporters. Okay? Stuff that is freely soluble in lipids, so is lipid soluble, and that is small, just crosses the plasma membrane. Okay? Things like oxygen, carbon dioxide, steroid hormones, fatty acids, okay, all just freely cross the plasma membrane. They're nonpolar and they're small. Okay, so we're going to talk about assisted membrane transport, and we're going to call it diffusion. Okay, and then we're going to talk about assisted membrane transport, and that just involves transporters. Okay, and we've got carrier-mediated transport, where we've got things that diffuse through channels. So water diffuses through channels because water's polar. And the diffusion of water is called osmosis. Right? And then ions, when they move across the plasma membrane, are moving through ion channels. Okay, so when they're moving down their electrochemical gradients, ions move through ion channels. Okay, larger molecules like glucose and amino acids move by what's called facilitated transport. Okay, so all three of these, osmosis, movement of ions through ion channels, and facilitated transport, is all moving things down their gradients, right? And doesn't require the use of energy. And then we'll talk about active transport. Active transport is moving substances against their concentration gradient. So you can think of it as uphill, right? And it requires energy. And then the last type of transport is vesicular transport. Okay, and that's endo or exocytosis.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. Cells use all these processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes.
Okay, so we're going to talk about unassisted membrane transport first. So simple diffusion. So these are molecules just freely moving through the plasma membrane. Okay, and it's due to uniform spreading out, right? Because they're going to bump up against one another where they're in higher concentration. And so when they're in higher concentration, they bump into each other more frequently, and that drives them to where they're in lower concentration. Okay, so you can think of molecules not liking to be crowded. They want to spread out to where they're in lower concentration. So they're moving down their concentration gradients. And arrows have been shown, but I really like to use a triangle to demonstrate concentration gradients. So this is where it would be high. Okay, the base of the triangle, and then you have low concentration at the point of the triangle. And so when you're going down the concentration gradient, or sometimes we'll say you're going with the concentration gradient, okay, it doesn't require energy. Just like when you bike downhill, it doesn't require energy. Okay, and then when you go up the concentration gradient, or sometimes we say against, it's like biking uphill. It takes energy, right? So when molecules move down the concentration gradient, right, from where they're in higher concentration, which is in side 1 to side 2, this does not require energy. Okay, there's a number of factors that affect how quickly a molecule is going to diffuse. And those factors get summarized in what's called Fick's law of diffusion. Okay? And factors that affect diffusion rate, or net flux, okay, so net flux and diffusion rate are synonyms, are the concentration gradient of the substance, that's delta C. The permeability of the membrane to the substance, that's P. And the surface area of the membrane, that's A. Okay, these are all in the numerator. So when they increase, what happens to net flux? It increases as well. Okay, so the greater the concentration gradient, the faster the diffusion rate. So the more of a substance you pour on one side, the faster the diffusion rate is. So that's the idea behind putting someone on supplemental oxygen. Right? You put more oxygen on the alveolar side, and that's going to increase diffusion rate into the bloodstream. Okay, permeability of a membrane to a substance. What is the membrane naturally permeable to? What?
Oxygen.
Toxins?
Oxygen.
Oh, oxygen. It's like, yeah, sometimes toxins, that's true. Oxygen, so lipid soluble or nonpolar substances. Okay, you don't really alter your membrane fluidity quickly or much at all, right? So this is sort of stationary. It's just substances that are permeable in the membrane, can freely cross, and will cross more quickly. Okay? Surface area of the membrane. The bigger the surface area, the faster the diffusion rate. Okay? So capillaries are highly branched. So the surface area of your capillary beds is the largest of your vasculature. So you have way more capillary surface area than you do for your veins or your arteries or even your arterioles. Okay, because that increase in surface area allows diffusion to occur faster. So oxygen can leave your capillaries and enter your cells. Okay, oh, and then cells that are specialized for absorption. Remember we talked about those microvilli? That increases the surface area of the plasma membrane, increases diffusion rate. Okay, so anything with microvilli is specialized for absorption or to maximize diffusion rates. Okay, in the denominator, we have the molecular weight of the substance and the diffusion distance. So as something in the denominator increases, what happens to the diffusion rate? It decreases. So as the denominator gets bigger, the answer gets smaller, right? They're inversely related. Okay, the molecular weight of the substance is fixed as well. This means small substances diffuse faster than large substances. But once you change the molecular weight of something, it's no longer that thing, right? Okay, so oxygen and carbon dioxide, which one is smaller? Oxygen, right? And so it has a faster diffusion rate because it's smaller. Okay, diffusion distance. You want to minimize diffusion distance. Okay, so this is why the alveoli in our lungs are just one cell thick, and those cells are really flattened. Okay, to minimize diffusion distance into the capillary beds, which are also an endothelial, flattened endothelial cell thick. Okay. So you can memorize this equation, or you can just think through it. So if a concentration gradient increases, what's going to happen to the diffusion rate? Right? It's going to increase. Right? If diffusion distance increases, what's going to happen to the diffusion rate? It's going to decrease. Okay? And that can come from memorizing that equation and putting it on your little cheat sheet, right? Once you get your blank scratch paper, or it's just from intuitively thinking through it. Okay. Questions about simple diffusion? Okay. Yeah.
There won't be actual
Numbers. There won't be actual numbers that you have to calculate. No. I won't give you a concentration gradient and a permeability coefficient and blah, 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 and make you calculate the diffusion rate. No. Okay. No need for a calculator on the exam. Okay, so now we're going to talk about assisted membrane transport. And so we'll still talk about diffusion, because diffusion describes any time a substance moves from high to low concentration. Right? But now we're going to talk about carrier mediated. And so we're going to talk about diffusion through channels. Okay, facilitated diffusion, where we actually have transporters. And then we'll talk about active transport. And active transport requires energy. So that's moving things up the concentration gradient. Okay, carrier-mediated transport is for molecules that are too large and or polar to get through the lipid bilayer unaided, right? They need a transmembrane protein to form a channel or a pore or to actually bind and bring it into the cell or bind and pump it out of the cell. Right? And carrier-mediated is either active or passive. And it's all about which way the molecule is moving. If it's moving down or with its concentration gradient, it's passive. If it's moving up or against its concentration gradient, it's active. Okay? An active transport requires energy input. And then we'll talk about vesicular transport, which is bulk transport. So endocytosis bringing molecules in bulk, exocytosis releasing molecules out of the cell in bulk. Okay, let's first talk about osmosis, because osmosis is obviously very important. Okay, so osmosis is the diffusion of water. And diffusion of water is water moving down its concentration gradient. However, water cares about other molecules. Okay, glucose only cares about glucose. If it diffuses down its concentration gradient, it diffuses from where there's more glucose to where there's less glucose. Water is going to diffuse from where there's more water to where there's less water. And where there's less water, there's more solutes. Right? And solutes are anything that dissolves in water. Okay, so oxygen's not a solute. Lipids are not solutes. Okay, glucose is a solute. All the ions are solutes. Okay, so solutes are anything that dissolves in water. And so water is going to move from where it's in higher concentration, where there are fewer solutes, to where it's in lower concentration, which is where there's more solutes. So I also like to say water wants to be where the party's at, which is where all the solutes are. Okay, most cells have aquaporons, or pores, in the plasma membrane to allow water to freely pass and do osmosis. Okay, most cells, not all cells. But most of your cells are freely permeable to water because they have aquaporons. Okay, so here we have a beaker that has a membrane, okay, and we've got water being shown in these blue-green circles and solutes being shown in purple. Okay, so this side of the membrane has more water and fewer solutes than this side of the membrane. So water is going to diffuse down its concentration gradient to where there are more solutes. Okay, so here we have a cell with a phospholipid bilayer. Okay, and this cell has an intracellular osmolality of 300 milliosmoles, which is normal, right? So there's 300 milliosmoles of solutes inside the cell. And we dump that cell into pure water or deionized water. Okay, and that water has an osmolality of 0. No solutes in it. So which way is water going to move? Into the cell. And what's going to happen to that poor cell? It's going to burst or lyse. Okay? So if you put your cells into deionized water, they will rupture or lyse or burst. Okay? Here we have a cell. Intracellular fluid is 300 milliosmoles. Okay, and we put it into a highly concentrated solution that's at 1,000 milliosmoles. Okay, so this solution is hypertonic. Higher concentration of solutes in the solution than inside of our cell. Water moves out. What's going to happen to that cell? It's going to shrink, shrivel, or crenate. Okay? So this is what happens when you get dehydrated. Your extracellular fluid's becoming highly concentrated. Okay? So water leaves your cells. Okay, so tonicity, I used the term hypertonic already. Right? That's a measure of tonicity. So all about that water gradient across of cells. You want to keep your cells in an isotonic solution. And isotonic means solutes in equals solutes out. Our cells want to be at 300 milliosmoles. So that means we need to maintain extracellular fluid at 300 milliosmoles. Right. I have a question for you. Are the solutes the same in the intracellular fluid versus extracellular fluid? No, right? It's the same concentration of solutes, but the makeup of the solutes differs greatly. Okay, inside of cells, we've got a whole bunch of protein and lots of potassium. Outside of cells, we have loads of sodium and chloride and some calcium. Okay, it's all just about numbers of solutes, not the actual makeup of the solutes. Okay, so here we have red blood cells in an isotonic solution. Solutes in equals solutes out. We get a net flux of water that's 0. We keep our red blood cells nice and plump. Okay, and you did this in lab, right? When you put your red blood cells into the hypertonic solution, where you had more solutes in the solution than inside of the cells, so the tonicity of the extracellular fluid was increased, right? Water moved out and your cells shrunk. If you put your cells into a hypotonic solution like deionized water, right? There's very little solutes outside, way more solutes inside. That pulls water towards them. Cell swells and will eventually rupture or lyse. Okay? So this cell has crenated. This cell is about to lyse. Okay, so we regulate extracellular fluid composition to keep our cells happy. What organs play a major role in regulating extracellular fluid composition? Kidneys. Yeah, exactly. Okay, so osmolality is the amount of, or the osmoles of the solute per kilogram of solvent. And we're talking about water being our solvent. So 300 milliosmoles means there's 300 milliosmoles of solute. And that solute might be sodium chloride and calcium. Okay? Doesn't have to be a single thing. It's just any solute in a kilogram of water. Right? And we are tightly regulating the osmolality of our extracellular fluid so that our cells can regulate the intracellular fluid. Right? And we have a negative feedback mechanism to do this, one of which is antidiuretic hormone. Okay, so in our hypothalamus, we have osmoreceptors. Okay, so these are receptors that can detect the osmotic pressure of our extracellular fluid. Where's the hypothalamus? It's in the brain. Okay, it's the only part of the brain not protected by the blood-brain barrier. One of the reasons is so that it can detect the osmolality of your bloodstream. Okay, and what it does is when it detects that the osmolality is increasing because you're becoming dehydrated, right? It tells the pituitary gland to release antidiuretic hormone, right? Which is ADH, which is also known as vasopressin. Okay, so the pituitary gland releases antidiuretic hormone, which then acts on the kidneys and tells the kidneys to reabsorb more water so that you produce low volume, highly concentrated urine. Okay, antidiuretic hormone also causes you to be thirsty, so you drink water, right, and replenish your water stores. So here we've got a negative feedback diagram. So dehydration decreases blood volume, increases the osmolality of your plasma. Hypothalamus detects that, causes antidiuretic hormone secretion, tells the kidneys to increase water retention, also causes you to be thirsty, which makes you drink, which increases water intake, all of which negatively feeds back. That's what this little negative sign is for. So that you increase blood volume, you decrease plasma osmolality, and you shut off the response. That's the negative feedback. You shut off the response. You stop producing antidiuretic hormone. Okay, so you're thirsty, you chug a bunch of water. Now maybe your osmolality drops too much. Okay, so you want to shut off antidiuretic hormone secretion so that you'll produce high volume, low concentrated urine. Right? Any questions about osmosis? Okay, let's talk about diffusion through ion channels. Okay, so ion channels are these transmembrane proteins. So here we've got the phospholipid bilayer, and they're forming a pore within that membrane. Okay, they can either be gated, so they open or close to do some sort of stimuli, or they're always open. And when they're always open, they're called leak channels. Okay, and all of our cells have loads of leak channels for what ion? Potassium. Okay, and that ion channel is sized just perfectly for potassium. Only allows potassium through. Potassium's only going to move down its concentration gradient. Okay, so when you hear the term ion channel, ions only move down their electrochemical gradients through ion channels. Okay, so ion channels are only for diffusion, so for ions to move down their electrochemical gradients. Okay, the factors that affect the rate of transport, okay, is that each ion channel has a particular transport rate. It only allows so many potassium through, right, in a specific amount of time. Right, maybe they have to squeeze through so it's slighter, or so it's longer. They have to squeeze through the ion channel so it's a longer process, or maybe the channel is slightly larger and so a bunch can go through all at once. Okay, and then the other thing that affects rate of transport is the number of channels in the membrane. So the more channels the membrane has, the faster the diffusion rate. So that's what that video was talking about. So we have way more potassium leak channels in the plasma membranes than sodium. So the diffusion rate of potassium is much faster than the diffusion rate of sodium. And so we say our cells are permeable to potassium at rest because the diffusion rate is faster because we have way more channels.
With potassium,
Yeah.
you said that the chemical gradient's stronger, right?
Mm-hmm.
So why do we have more potassium in the center? Like I can see that the electrical gradient wants it in, and it can pass freely. So I don't understand that.
Okay, who wants to answer this question? Why do we have more potassium inside our cells?
In part because of the, was it sodium potassium pump?
Exactly, the sodium potassium pump.
Yeah, so we're pumping sodium out while we pump potassium in.
Yeah.
As well as there is an electrical, actually, never mind, I might be wrong on that one.
No, I think you're right. Keep saying what you want to say.
There is an electrical gradient pulling the potassium in, which slows the diffusion out.
Yeah, no, exactly. Okay, so we expend energy to continuously pump potassium back into our cells. Yeah, so it diffuses out and then we pump it right back in. And then it diffuses out and then we pump it right back in. Yeah, Okay. And then also that electrical gradient holds some of it in because it has an opposing chemical and electrical gradient. Okay. Any other questions? Right. So let's talk about facilitated diffusion. Right, so facilitated diffusion, it's for substances that are larger than ions and water. So things like glucose and amino acids move by facilitated diffusion. Facilitated diffusion is moving a substance down its concentration gradient or with its concentration gradient because it's a form of diffusion. And it's passive. Right? Yes?
Is facilitated diffusion for ions or particles that are both polar and nonpolar?
Well, how do nonpolar molecules move through the plasma membrane?
They just diffuse through.
They just diffuse through. Yep, so the facilitated means there's a protein involved. Okay? Yeah.
Well, what about glucose? Because I don't think glucose has a charge, so theoretically it should be able to just diffuse right through.
Oh, you're right. Glucose does not have a charge, but is it polar? What is the functional group all over carbohydrates that make them polar? Who remembers the functional group from that table? What? Hydroxyl groups, exactly. Carbohydrates have hydroxyl groups that make them polar. Okay, so anything, this is how you can think of it. Anything that dissolves in water is polar and therefore is not going to freely cross the plasma membrane. Anything charged is polar. right? But there's lots of uncharged molecules, like carbohydrates, that are polar. And carbohydrates are polar because of their hydroxyl groups. Okay? Amino acids are polar, but they often become charged because they get rid of a hydrogen ion. Yeah?
So, do the carrier proteins use energy?
No. For facilitated diffusion. We're going to talk about carrier proteins for active transport, and those use energy. Okay? But if I'm talking about facilitated diffusion, this is for molecules that are larger than ions like sodium and potassium or water, but they're too polar to cross the membrane themselves. So they have to have a protein carrier involved. But because it's diffusion and it's moving a substance down its concentration gradient, because that substance wants to move in that direction, energy is not required. Right? So here we have an example of how glucose enters our cells once glucose gets absorbed from the small intestine. So we'll talk about glucose absorption from the small intestine. That's active, which we saw in that video. Right? But once it's in the bloodstream, glucose is going to move down its concentration gradient. So this is how it enters cells. Okay, so it enters cells by facilitated diffusion. So there's a protein transporter, which is open to the extracellular fluid. Okay, so we have less glucose inside of cells than outside of cells. What happens to glucose once it enters our cells? Why is it in lower concentration? What do we use it for? ATP production, right? We clip it apart. Once we clip it apart, it's not glucose anymore. So more wants to come in. Okay? Or we store it as glycogen. Once it's glycogen, again, more glucose can enter. Okay, once glucose binds to the transporter molecule, it actually flips and opens to the interior of the cell. Okay, so glucose binding provides the energy to flip and open to the interior of the cell. Okay, these are called permeases. Right? And they're common for things like glucose and amino acids. Right? And they'll move those substances from where they're in higher concentration to lower concentration. Hey, how do we know if something's moving by facilitated diffusion versus simple diffusion? Right? We would have to do an experiment, and what we would do is look at the net flux of the substance when we added more and more of it. Okay, so this is the concentration of, let's say, glucose. Okay, so we want to know, is glucose passively going across the plasma membrane, or is it moving by a transporter? And so what we do is we have some sort of membrane, we're looking at it moving from one side to the other, so that's net flux, and then we just pour on more and more glucose. Right? If glucose was moving by just simple diffusion, this line would be straight. It would be straight and it would just go on forever. Okay? If it just can diffuse through the plasma membrane. Because it's being transported, we get an asymptote, or it flattens out. Right? And it flattens out because carriers display saturation. So basically, all the carriers on that plasma membrane are transporting glucose as fast as they can. And they can't transport it any faster, so they become saturated. So no matter how much more glucose you pour on, more is not being absorbed faster. Right? How could a cell increase the diffusion rate? Put more transporters on. Right? Okay. Or if we wanted to decrease the diffusion rate, it could put fewer transporters on. So this is what happens in your kidneys, right? So your kidneys are trying to reabsorb any glucose that gets filtered out, but because it's transported, it displays saturation. So if you start spilling glucose into your urine, it means blood glucose levels are way too high. Right? Okay, everyone fine with passive forms of transport? All of them are moving substances down or with their concentration gradient. Energy is not required. Okay, we're now going to switch and talk about active transport. This is moving substance against their concentration gradient or up their concentration gradient. Okay, and you can think of it as moving something uphill. You always have to have a carrier molecule. Okay? This means that nonpolar substances that freely cross plasma membranes can never be actively transported. So our cells can never actively transport oxygen into them. Right? You always have to have a higher concentration of oxygen outside of your cells in order to get oxygen to enter your cells. Right? Because oxygen can never be actively transported. Molecules that are polar can get actively transported, right? Because they require a carrier molecule. And then we're going to separate active transport into two types, primary and secondary. Okay, primary requires the direct use of ATP. So these transporters usually have a name like blank blank ATPase. And that ATPase means that that transporter has an enzymatic portion that clips that third phosphate group off of ATP. So anything that's blank blank ATPase is a primary active transporter, right? It directly uses ATP. The transporter itself cleaves ATP. Secondary active transport are driven by gradients set up by primary active transporters. Okay, so primary active transporters are setting up ion gradients, right? And we set an ion gradient up on a cell. So we've got higher sodium outside of our cells, higher chloride outside of our cells, higher potassium inside our cells, higher calcium outside. So what a secondary active transporter is going to do is oftentimes it uses sodium. And so sodium really wants to get in our cells. And so the secondary active transporter will allow sodium to enter the cell, and that will drive the transport of a second molecule against its concentration gradient. Okay? And the video talked about an active transporter for glucose. So in our intestines, glucose is in higher concentration in those epithelial cells lining the intestine. So to absorb the glucose, we let sodium in, and that pulls the glucose with it, because sodium so badly wants inside the cell. Okay? Primary directly uses ATP. Secondary uses an ion gradient set up by a primary. Okay? So energy is involved. Okay, so primary active transport is how our cells pump calcium out of them. Right? So this is how our cells set up the calcium gradient. Right? Where we have low calcium inside, high calcium outside. Our cells expend energy to set up that gradient, and they use calcium ATPases or calcium pump. Okay? So we've got low calcium inside our cell. It's going to bind to one of the calcium ATPases. That ATPase is actually going to cleave that phosphate group off of ATP and use the energy from that exergonic reaction to pump calcium out, to pump calcium where it doesn't want to be. Okay. The sodium-potassium pump, which is also known as the sodium-potassium ATPase, is how our cells set up the sodium and potassium gradients. And we've already talked about it a few times, right? And I'm going to add it to my cell as a star. And then I'm going to put 3 sodium out and 2 potassium in. Okay, so the sodium-potassium pumps, which are present on all our cells, right? All our cells have many, many sodium-potassium pumps. Okay, they are constantly working and constantly using ATP to pump 3 sodium out. So they have a binding site for 3 sodium molecules. So they're open to the intracellular fluid. They bind 3 sodium. And then they clip that phosphate group off of ATP. And they use the energy from that reaction to pump the sodium up their electrochemical gradient. Okay, and then they're going to bind 2 potassium from extracellular fluid. And then they'll release that phosphate group, and that will provide the energy to pump the 2 potassium into the cell where they don't want to be. Okay, and notice they're pumping 3 cations out for every 2 they pump in. So again, that is why net negative charge on the interior of the cell. Okay, this pump is not electroneutral. It pumps out one extra cation. Okay, so it helps to set up the resting membrane potential. Which is why I mentioned it multiple times before we even officially talked about it, right? And it was in that video about resting membrane potential. So all our cells have these resting, or have these sodium-potassium pumps. All right, so we talked about primary active transport. In primary active transport, those transporters directly use ATP to move molecules up or against their concentration gradients. Okay, again, if you draw a concentration gradient as a giant triangle, right, the top being where it's in high, and the point being where it's in low, okay, when you go down the concentration gradient or with it, does not require energy because it's a downhill movement. Okay? When you go up or against it, it requires energy. Primary active transporters directly use ATP. They clip a phosphate group off of ATP, and that provides the energy to move something against its concentration gradient. Secondary active transporters use an ion gradient set up by a primary active transporter. Right? So here we have an example of using sodium. So what's the primary active transporter that sets up the sodium gradient? The sodium-potassium pump. Okay, so the sodium-potassium pump is a primary active transporter. It sets up that sodium gradient where we have high sodium outside the cells, low sodium inside the cells. This means sodium really wants into our cells. Okay, and letting it into the cell provides this transporter, which is a secondary active transporter, with the energy to pull glucose up its concentration gradient. Right? So it's not directly using ATP, it's indirectly using ATP. Okay? So this transporter, which would be called SGLT1, which is the sodium-dependent glucose transporter 1, meaning the first that was discovered. That's where the number comes from. It only runs if the sodium-potassium pumps run. Okay? The sodium-potassium pumps are disabled. The SGLT1 transporter won't function. Okay, this is called co-transport because both molecules are moving in the same direction. Okay, so secondary active transporters that transport the molecules in the same direction are co-transporters. In panel B, we have counter-transport. Okay, so by allowing sodium into the cell, that gives this hydrogen, the sodium-hydrogen counter-transporter, the energy it needs to pump hydrogen ions out of the cell. Right? And why do cells want to pump hydrogen ions out? Acidity. Okay, if you have a buildup of hydrogen ions inside the cell, that's going to lower the pH. When you lower the pH, that affects what molecules inside your cells? Proteins. It will cause them to denature. Right? So your cell is allowing sodium into it in order to pump hydrogen ions out, right? To maintain a neutral pH. So this is how sodium gets into the cell. So if you're wondering where does the sodium-potassium pump get those 3 sodiums to continuously pump out, it's because sodium is allowed in due to secondary active transporters. Okay, so membrane potential serves as a source of potential energy. And one of the ways those cells use that potential energy is to do secondary active transport. And then next week, we'll talk about another way to send signals rapidly. Okay, so action potentials are a way to use membrane potential as well. All right, so here we have some cells, some epithelial cells, where they have microvilli. And what are those microvilli telling you? Increasing surface area. So these are epithelial cells that are specialized for absorption, right? Because the greater the surface area, does that increase or decrease diffusion rate? Increases diffusion rate. Okay, bringing all sorts of concepts together right here. Okay, so these are very similar, or are the cells that either line the small intestine. Okay, so the epithelial cells lining the small intestine are called enterocytes, and they have microvilli, and they're specialized for absorption. But we also have cells in the proximal tubule of our nephrons that look just like enterocytes, that have the microvilli, and they're specialized for absorption. And they're reabsorbing everything that got filtered out. Okay, so they act very similarly. We have a lumen, which is an external body surface. Okay, and then we have interstitial fluid. So we have two different membranes. This membrane is the apical membrane. Right, so that's facing the lumen, or the external body surface. And then the one that's facing the internal body surface is the basolateral membrane. Okay, so what you notice is we have different types of transporters on those two membranes. So if we look here in panel A, we have ion channels for sodium. So sodium is able to freely enter the cell from the lumen. Okay, so all that salt in your diet, super easily absorbed. Okay, and then on the basolateral side, we have the sodium-potassium pumps. Okay, so enterocytes have tons of sodium-potassium pumps, as do proximal tubule cells, but they're only on the basolateral side. Okay, so they're pumping 3 sodium out into the interstitial fluid, and then 2 potassium in, and then you only have potassium leak channels on the basolateral side as well. Okay, so we're setting up a sodium-potassium gradient. So an enterocyte has a resting membrane potential that's identical to all your other cells. High sodium outside, low sodium inside, high potassium inside, low potassium outside. Okay, here we're adding a few other transporters. Okay, we're going to add the molecule glucose. So that's this green hexagon. There is more glucose inside your enterocytes than in the lumen. Because what do you eat most of your sugar? What form is it in? Well, your carbohydrates. Sorry, I don't mean sugar. What form is your carbohydrate in? What are you supposed to eat it as? No, not as sugar. You're not supposed to eat a bunch of sugar. Complex carbohydrates, so long chains of glucose. Okay, so you have complex carbohydrates hanging out in the lumen of your intestine. Okay, the enzymes that clip them into just glucose monomers actually sit on that apical membrane. Okay, so as soon as glucose gets formed, okay, it gets absorbed. So that means you actually have pretty low glucose levels in the lumen. Unless you're eating a boatload of pure glucose, which, again, if you even eat a lot of sugar, most of it's disaccharides, right? In the form of sucrose or high fructose corn syrup. Okay, that's still a lot of sucrose in there. Yeah. Okay. So there's low glucose in the lumen, right? High inside the cells. So that means it has to be transported actively. So that's why it's getting a red arrow. Okay, and we just talked about this is SGLT1, that sodium-dependent glucose transporter. It is a secondary active transporter. It lets sodium in down its electrochemical gradient in order to pull glucose up its concentration gradient. Okay, glucose is not charged. That's why I said just concentration gradient. Now we have loads of glucose inside the cell. Right? And as soon as it gets transported out of the cell, it gets picked up by the bloodstream. So that means there's less glucose in the interstitial fluid than in the enterocytes. So now it's energetically favorable for glucose to leave the cell. It wants to leave the cell. So that means the transporter for glucose on the basolateral side can be what type of transporter? Is it going to be active? No, it's not going to be active. So how would glucose be transported in a non-active way? Is it going to just diffuse through the membrane? No. I heard the answer. Facilitated diffusion. Okay? There's no ion channel because glucose isn't an ion. There's no glucose poron, right? Like there are aquaporons for water. It's going to go by facilitated diffusion, and that facilitated is by that transporter, right? And those transporters are called GLUT. So they're GLUT. And those transporters. Okay, so it moves by facilitated diffusion across the basolateral membrane. Yeah.
Is it safe to say then if you have diabetes and the glucose is staying in your blood that these cells can really take a hit because the glucose is staying, it's not moving?
Oh, interesting. Okay, so the question is if you are diabetic and you have super high blood glucose levels, does it ever become energetically unfavorable to absorb more glucose, and does it basically get trapped inside your enterocytes? If you're eating boatloads of sugar, maybe it's possible,
I know that
right? But you're also going to be starting to spill some of that glucose into your urine, right? And if you eat sugar, you're still going to be absorbing it.
I know that there's one of the things that I've found out with a lot of diabetics is that there's almost like an addiction to sugar. So they're constantly
Yes, but that's your brain.
right.
Yeah. So the sugar addiction is because our brain desperately wants glucose, and that's why sugar is delicious to us, right? And you can actually have a down regulation of the number of sweet receptors on your tongue, so things don't taste as sweet to you anymore. So you actually want more, right? So there is sort of, and the lights off the dopamine pathways, which also get lit for other things that are considered addictive. Yeah. That's not necessarily due to the transport in the small intestine. These GLUT transporters are the ones that get put on cells that are insulin sensitive, though. Right? So the way that your insulin sensitive cells, so your liver and your skeletal muscle and your adipocytes, the way that they absorb glucose is that when insulin binds to the receptor, they put those facilitated diffusion transporters on their membrane. So they're only going to absorb glucose when insulin's in the bloodstream, right? When insulin's not in the bloodstream, they take those transporters off, right? And they won't absorb any glucose. It stays in the bloodstream for your brain. Okay? Your brain always has those transporters on those capillary cells. Okay, so it's going to absorb glucose no matter what. It doesn't need insulin in the blood or glucagon in the blood, right? But when you have way too much blood glucose, the reason you spill it into your urine is because you only have so many SGLT1s on the apical membrane of those proximal tubule cells. Okay, and once they get saturated, glucose is going to stay behind, ends up in the urine. Right. Okay, so we have a secondary active transporter here. We've got facilitated diffusion here. We've got an ion leak channel here for its potassium. And we have a primary active transporter, the sodium-potassium pump, being shown here. Okay, so a nice summary of all the different types of assisted transporters. Right? What's an example of a molecule that will be absorbed by simple diffusion from the intestine? So it'll just diffuse through the plasma membrane. What's going to just diffuse through?
Non-polar small.
Nonpolar small. Thank you. So something like fatty acids will just diffuse right through. Okay, we don't use our gut for absorbing oxygen, right? What organ system do we use for absorbing oxygen? I don't recommend swallowing a bunch of air because what will happen? How will it come out? One of two ways. You're either going to burp, right? Or you'll have flatulence. Okay. Okay. Any questions about assisted transport? And this figure does a nice job of summarizing it. Last type of transport we'll talk about is vesicular. So this is moving bulk. So bulk moving. This is the Costco of transport. Okay. So material gets moved into or out of the cell, wrapped in a membrane. Okay, so you have bulk materials wrapped in a membrane that fuse with the plasma membrane, then get released to extracellular fluid. That's exocytosis. Okay, and then endocytosis is you bring things in bulk and wrap them in a membrane. Okay, so endo means bringing into the cell, exo means removing from the cell. And we'll talk very briefly about the 3 types of endocytosis. So pinocytosis is known as cell drinking, so dissolved molecules. Phagocytosis is known as cell eating, so large molecules, something like a bacteria. Okay. And then receptor-mediated endocytosis just means receptors are involved. Okay, so let's look at endocytosis first. Okay, so here we have a nice cartoon showing all 3 types. So here's phagocytosis. Our phagocytic white blood cells do a load of phagocytosis. And who knows which are our phagocytic white blood cells?
White blood cells.
Well, not all white blood cells are phagocytes. Monocytes, once they turn into macrophages. Okay, so monocytes are immature macrophages. The macrophages hang out in our tissues and they phagocytize all sorts of stuff. Okay, it might be bacteria, it might be worn out cells, etc. Okay, what are the phagocytes that stay in our bloodstream? Starts with an N. Neutrophils. Okay, so there are phagocytic specialists that hang out in the bloodstream. Okay, so what they're doing is they are engulfing particles, or you could think of this as a bacterial cell. So what they do is they actually wrap that bacterial cell in part of their own plasma membrane, and then it pinches off. Once it pinches off, it's called a phagosome. Okay, so it's surrounded by a lipid bilayer. And then generally what happens is phagosomes fuse with lysosomes. And what are lysosomes filled with? No. It does start with hydro. The peroxisomes are filled with hydrogen peroxide, or they're detoxifying hydrogen peroxide. What do lysosomes have? No, not hydrochloric acid. But good guess, because I said hydro, right? Hydrolytic enzymes, right? What's hydrolysis?
Putting water in.
Putting water in order to break bonds. Exactly. So lysosomes are filled with hydrolytic enzymes that break things apart. Okay, so then the lysosome will digest that bacteria, and then the cell can actually use all the amino acids, carbohydrates, etc. that were present in that bacteria. If we're talking about white blood cells, they'll usually put a portion of the bacteria on their plasma membrane in order to warn other white blood cells. We won't talk about that until the immune system, way at the end of the semester though. Okay, but phagocytic specialists do a lot of phagocytosis, cell eating. Okay, pinocytosis is where you have dissolved molecules. They get wrapped in the plasma membrane, they pinch off, and then they get called endosomes. Right? Receptor mediated. Your book goes into a lot more detail about this. Don't worry about the details. Basically, what it means is that there's a receptor involved. So you have these receptors. Once they bind to the molecule, that tells the cell to form what's called a coated pit, and then it basically forms an endosome. Okay? With a lot of steps involved. Right? Don't worry about the steps. Okay? Just know the major difference between phagocytosis and pinocytosis. Right? And receptor mediated is a form of pinocytosis. A specialized form. Okay, let's talk about exocytosis. So exocytosis, we have a secretory vesicle that is filled with molecules the cell wants to release to the extracellular fluid, right? And next week we're going to talk about neurons, and this is how neurons release all their neurotransmitters. Right? So those neurotransmitters are all water soluble, okay? Because only water soluble stuff can get wrapped in a membrane and be trapped inside, okay? So we've got neurotransmitters. Think of these little orange circles as neurotransmitter, right? These secretory vesicles fuse with the plasma membrane and then open to the extracellular side and release their contents. Okay, this is also how beta cells release insulin. Right? Insulin's a peptide hormone. All peptide hormones get made in what organelle? What organelle is going to make proteins for secretion? The rough endoplasmic reticulum. And then what organelle is going to make those secretory vesicles? The Golgi, right? The Golgi. So the smooth is going to wrap the insulin into a vesicle that will then fuse with the Golgi, which will then release the vesicle that will fuse with the plasma membrane and release those peptide hormones to the extracellular fluid. Okay, sometimes you get transcytosis, okay, where you have endocytosis on one side of the cell and you have a vesicle that actually moves through the cell and then the contents get released by exocytosis on the other side. This is how a lot of clotting factors move from the bloodstream, right, into interstitial fluid. So you have clotting factors in your interstitial fluid. Those clotting factors get made in the liver, travel in the bloodstream. Endothelial cells in your capillaries will do endocytosis on one side, exocytosis on the other side. Right? Yeah.
The microtubules, those are the little like train tracks things.
Yes, exactly.
Do the endosome and the secretory vessel, do they travel along those as well?
Definitely.
Okay.
Yep. Okay, so bringing it all the way back to the cytoskeleton, remember those microtubular elements form the train tracks, right? And then there's motor proteins that walk along them. They're going to be pulling those secretory vesicles or those endosomes or the phagosomes to their proper location. Okay, so it's not like you form a vesicle and then it just floats around in your cell, right? It gets directed somewhere. Okay, any other questions? Yeah.
So I just want to make sure I understand this right. So it's taken in, the particle is taken in,
Yeah.
and nothing happens as it passes through the cell, and it's just kicked out on the other side.
That's what it's showing for this.
Sounds good.
Yep, that's what it's showing for this epithelial cell. Right. I don't know how often this actually occurs in enterocytes. Right. I know it's a well-known fact that it occurs in capillary cells. That's how clotting factors get across. All right. Any questions about membrane transport? Yeah.
So, could you briefly touch on again what's the difference between pinocytosis and phagocytosis?
Okay, so the difference between pinocytosis and phagocytosis. Phagocytosis is bringing in large particles, so it's known as cell eating. Okay, pinocytosis is bringing in dissolved molecules, so it's known as cell drinking. Right, so dissolved molecules are much smaller. You're just bringing in a whole bunch at once, whereas phagocytosis, you have this large particle or another cell that you're trying to bring in all at once. Yep, okay. Any other questions about membrane transport? Right.